We now turn to the 2016 Jacques Miller Medal for Experimental Biomedicine. This is again the second time this medal has been awarded. The Jacques Miller Medal recognizes research of the highest standing in the field of experimental biology research and is made in honor of the contributions made to science by Professor Jacques Francis Albert Pierre Miller, a Fellow of the Academy. The recipient of this year's 2016 Jacques Miller Medal is Associate Professor Katherine Kedzierska.
Dr. Kedzierska combines cutting-edge basic research with unique clinical studies to define how to generate protective immunity against pandemic and newly emerged influenza viruses. Her research identifies key factors that drive the severe and fatal influenza disease in high-risk groups, including the young, elderly, pregnant women, immunosuppressed individuals, and Indigenous Australians. Her findings on the optimal human immunity to influenza viruses have implications for vaccine design and development and are applicable to other infectious diseases and tumors. I invite you to address us on the immunity to pandemic and newly emerged influenza viruses.
Thank you. And I'd also like to thank the Academy for awarding me the Jacques Miller Medal. I'm greatly honored, especially as Jacques is such an icon in immunology in Australia and internationally. So we study immunity to influenza viruses, as influenza poses 3 global health challenges. The first one, as you know, these are our annual seasonal epidemics causing great morbidity and mortality, especially in children and elderly. Then when the virus reasserts, we can have an outbreak of influenza pandemic, and then virus spreads globally and causes millions of infections. Virus also infects animals and now and then jumps the house and can cause really severe disease in humans. So while B cells and antibodies provide fantastic neutralizing immunity, this immunity is really strain-specific. On the other hand, killer CD8 T cells, which are the cells we are interested in, can provide broader immunity as they recognize peptides from the core of the virus that's specific again, that's the same in all the distinct influenza viruses. So our research interest is really to understand how we can generate and maintain influenza-specific CD8 T cell immunity. We dissect those CD8 T cells across distinct influenza strains that circulated over the last century and across different ethnicities. And we'd like to understand why some people are really at risk of influenza infections, especially patients, those hospitalized with severe influenza, young children and elderly, and Indigenous Australians. So as you remember, in 2009, we had an outbreak of the first influenza pandemic of the 21st century, swine-derived H1N1. It was the first cases were reported at the end of April. In under 2 months, the virus spread to 74 countries. So at the beginning of each influenza pandemic, there is a global panic, as you can see here in those, as exemplified by those headlines, as we do not know how virulent the virus is and how many deaths it will cause. The rapid spread was due mainly to minimal antibody recognition across the globe, and there was preferential infection of children and young adults, but elderly were not susceptible to the virus. So we asked the question if at the population level we have minimal antibody immunity, do we have any of the killer CD8 T cells that can provide us some level of protection towards the pandemic virus? And the answer was yes. There was quite a high 70% conservation level between the antigenic, T cell antigenic regions within the new virus and the viruses that, seasonal viruses that circulated that we would have been exposed to. And an interesting finding was that the 2009 H1N1, the T cell regions were quite similar, very similar to the regions from the catastrophic Spanish influenza in 1918 and the viruses that circulated at the beginning of the century, which partially would explain why elderly had more robust immunity to the new virus. Similarly, in 2013, we had an outbreak of a new avian-derived virus, H7N9. So it was reported, the first cases were reported in China in April. And now we had more than around 700 cases. The mortality rate is 40%, 99 hospitalization rates. The virus causes really severe acute pneumonia. So there are quite a few concerns. At the population level, we have no neutralizing antibodies. This is the first time we've seen H7N9 virus in human population. The virus is well established in birds. However, it causes only mild disease in birds, so we do not know which birds are sick, which birds we need to cull. And for now, it's only bird-to-human transmission. However, there is always a possibility of further mutations and that the virus can acquire human-to-human transmission. So obviously, it is important to understand how we can control the new virus. And we also dissected the killer CD8 T cell immunity, whether we would have any immunity to this novel virus. And the results were actually quite interesting, as we found that pre-existing CD8 T cell immunity varies greatly across ethnicities. So the highest population coverage, around 60%, is in Caucasians, then just above 35% in Oriental African Amerindian people, and we identify two populations who would be at risk if the virus spreads out of China, and that's Australian and Alaskan indigenous people. And that actually verified the previous findings that 10 to 20% of indigenous people died from the Spanish influenza, as well as the hospitalization levels were high in 2009. Similarly, indigenous Alaskans, up to 100% of adults died in some isolated villages. So we know there are ethnic differences in sensitivity to H7N9, but the important question was whether the killer CD8 T cells made any difference in patients who were hospitalized in China. So to answer this question, we worked with a fantastic collaborator, Professor Jianting Xu, and at the Shanghai Public Health Clinical Center, and two of my people, Zhang Fang, a postdoc, and Sergey, a PhD student, went to Shanghai and worked with Professor Xu's people to understand immunity to the new virus. During the first wave, they had 18 patients, 12 of them survived, 6 of them died, and to the best of our knowledge, these are the only longitudinal samples from patients in China. And because these are such unique samples, we set up many robust assays to give us as lots of information about immunity to the virus. So just to quickly summarize our findings, we found that yes, CD8 T cells made a difference, and patients with pre-existing CD8 T cell immunity had rapid recovery from the H7N9 virus and were released from the hospital within 18 days after infection. On the other hand, patients who were stay in hospital for longer, they had delayed CD8 T cell responses, had to recruit many different immune cell types. In contrast, patients who died had no cellular immunity at any stage of the disease. So obviously, now we're pursuing some of these findings, but what we are really interested in, why patients who died had no cellular immunity, what caused the total shutdown of this immunity. We're trying to identify biomarkers, early biomarkers that could tell clinicians which patients are at risk of dying, and obviously we are really interested in understanding why indigenous populations, especially indigenous Australians, are at risk from severe disease, especially when the new virus enters human circulation. So I just like to acknowledge everyone in my team. I'm really privileged to work with such a talented group of people who obviously did all the work. I'd like to acknowledge Peter Doherty, a fantastic and very inspiring mentor. For the last 14 years, we've got many wonderful collaborators, NHMRC and the University of Melbourne for funding and support. And thank you for your attention.
Thank you. Any questions?
Are the ethnic differences due to differences in exposure to viruses in the past, or are they genetic differences between the populations?
Well, I think both. So influenza viruses circulated in Europe for centuries. It was actually first described by Hippocrates, whereas influenza viruses are new to indigenous populations. So we know that Europeans co-evolved, had a long-term exposure to influenza viruses, co-evolved, MHCs co-evolved with viruses. We are looking at some differences in HLAs and T-cell responses that are restricted by those HLAs.
If there are no further questions, please join me in thanking Dr. Kedzierska.